in such a short period of time in college. Anyhow, so tonight we're going to start here with chapter 19, which is enzymes, and we're going to kind of get the ball rolling here, so to speak, so that you have a foundation to build upon in chapter 19, the chapter that deals with enzymes. So if you're wondering, what the heck is an enzyme? Well, earlier on in the class, we spent a lot of time talking about amino acids, right? If you remember an amino acid, an alpha amino acid, we said, okay, well, it's got an amino group, then it has an alpha carbon with a hydrogen, then it's got a carboxyl group over here, then it has some kind of R group that gives it its identity, so to speak. You could have a hydrophobic group or a hydrophilic group there, a basic or an acidic group there. So polar groups also as well. So we spoke about amino acids, and we said, well, actually, amino acids don't exist like that. They exist as Zwitter ions. But anyhow, we also spent a lot of time putting amino acids together. If you remember, when we looked at proteins, okay, we talked about how to make a peptide, and we said, well, a protein is just a string of amino acids. You remember we had NCC, right? And we would just keep going. Okay, and we'd say NC carbonyl, and then NC carbonyl, and then we'd fill in everything else, the nitrogens, the carbons. We put in all of our R groups. And what did we say? We said that when we put enough amino acid residues together, it ceased to no longer be a peptide. Then it became a protein. And then we discussed, primary, secondary, tertiary, and quaternary structures of proteins. And so enzymes are nothing more than proteins, right? We spent a lot of time talking about what proteins are. Well, I'm here to tell you right now that we have a whole second chapter that deals with proteins, except they're a special type of proteins, and we call them enzymes. So they must be a protein with a special function. And the function of an enzyme is to serve as a biological catalyst for reactions in all living organisms, so plants and animals. And if you think back to General Chemistry 1, which you all took, Introduction to Chemistry 1, many of you took it here at PPCC, we said, okay, well, how does an enzyme work in that class? And we were talking about non-biological catalysts there, but we said the way that a catalyst increases the rate of a reaction is by lowering the activation energy. You might remember, again, from General Chemistry 1, we said, okay, well, if you have a reaction, so we'll just write here, a reaction that occurs over, a certain amount of time. Okay, so here's our reaction axis. If this is energy over here, let's say you have some starting materials and they've got to get over, some kind of activation energy in order to get down to the products. And we said, okay, well, this difference in energy, this hill they have to climb, that's called the activation energy. And all a catalyst does is it's not going to affect the equilibrium concentrations of the starting material and the product or reactants and product. What is it going to do? It's going to lower the activation energy. Okay, so it's just going to make a lower activation energy. Also, something that you probably learned in general chemistry is not only will it lower the activation energy, but it will provide an alternate pathway. So the way that we represent that is we say, okay, well, not only does it lower the activation energy, but it might have maybe 2 or 3 steps or something, right? It's going to have a different pathway. So our can increase the rate of reaction, from 10 to the power of 6 to 10 to the power of 12-fold, but remain unchanged themselves. So basically reactions that couldn't even occur in our bodies at physiological pH and at body temperature, 37 degrees Celsius, are made possible by enzymes. Enzymes are also very specific, right? If we have these biological catalysts, they're not just going to react with any old reactant, so to speak, and we'll discuss what reactants are, but they're not just going to react with anything in the body. They have to be very specific about what they react with. And what we're going to see is that enzymes will catalyze a certain type of reaction only, right? Or maybe even only a certain reaction. So there's certain enzymes in the body that literally catalyze one specific reaction and nothing else. And then there's some other enzymes that might be a little more broader in terms of the scope of what they can do, and they might metabolize more than one type of reaction. Now, nomenclature is not a huge section of this chapter, but it definitely is important. And you probably noticed if you've read the chapter or looked at any of the videos that I've posted that the names of most enzymes end with A's, okay? Like things like peptidase, lipase, hydrolase. You've probably heard of some other ones like sucrase. Um, you've probably heard of kinases if you studied biology before. So you see that when we have this suffix ase, you're generally going to be speaking about some kind of enzyme. Something else that we're going to see later on is that some enzymes need a little help. You know, the Beatles said, I get by with a little help from my friends. Well, what we're going to see is that some enzymes require what's called a cofactor or sometimes a coenzyme in order for the enzyme catalyzed reaction to occur. And a cofactor is oftentimes a metal ion, okay? Like, you know, a transition metal, like maybe copper 2 plus. And if you're wondering about that, we'll talk about that more later on. Or a coenzyme, which can be an organic molecule. So generally speaking, I'd say that coenzymes are organic molecules, whereas cofactors are usually things like metal ions. And again, these are specific cases where the enzyme itself is not sufficient to allow the reaction to occur. The enzyme also needs something else to help it perform its function, and that would be a cofactor or a coenzyme. So kind of a little summary of a lot of the things that we're going to look at in the enzymes chapter. And here is a ribbon structure of an enzyme. Now, I just took this off of the internet, and you can see that, you know, enzymes have very complex structures, right? Very complex structures. And you can see, you recognize, you know, these curls here. These are alpha helices, right? This is an alpha helix, and you can see several of those throughout the molecule. What else do you notice with this ribbon diagram? This ribbon diagram also has, if you look very carefully, you see all these lines here. This is actually the bond line structure, you know, that you can kind of see in here. Now, obviously, there's not enough detail to kind of, you know, make good sense out of it. You might be able to tell a few things if you know a lot about amino acids. But the idea is that enzymes have very complex structures, and you can see that there's something that's looks like it's, maybe I should get a different color here. Something here that looks like it's not part of the enzyme, right? You see there's some kind of molecule here that's highlighted in green. And when we're talking about substrates or reactants that react in enzymes, or enzyme cause the reactions that convert substrates or reactants to products, you can see that the substrate or reactant is much smaller in comparison to the enzyme. All right, well, let's get into it. And this is section 16.1, which deals with nomenclature and classification of enzymes. Enzymes are often classified by placing them in categories according to the reactions that they catalyze. And you can probably guess, you know, what kind of reactions most of these enzymes catalyze just by looking at them. For example, an oxidoreductase, these catalyze oxidation and reduction reactions, right? If you think about good old redox chemistry, right? Redox chemistry. Now, redox is a term that we mostly use in General Chemistry 1, but we talk about oxidation and reduction enough in organic chemistry too, right? We say, okay, well, oxidation is gaining oxygens or losing hydrogens, and reduction is the opposite. Next would be a transferase. A transferase is going to be an enzyme that transfers a group from one substrate or from the enzyme to the substrate. A hydrolase is obviously going to be something that does a hydrolysis. A ligase, oftentimes liases involve double bonds, so the formation of a double bond or the cleavage of a double bond. Isomerase, that one's pretty abundantly clear just by looking at the name. It has something to do with isomerization, and so the number of atoms doesn't change when an isomerase acts upon our starting material. It just is going to rearrange or change the structure. And then finally, a ligase. Ligases catalyze reactions that involve specific types of bonds. So specific types of bonds, and we'll probably look at some examples of those along the way. Well, let's first start by looking at oxidoreductases. So oxidoreductases catalyze redox reactions, and oxidoreductases can be broken down into two subcategories. We can have either oxidases or reductases. Now, you can probably guess that reductases catalyze reduction and oxidases catalyze oxidation, and you would be completely correct if you thought that. So here's a specific example, right, of an oxidoreductase. And the name of the oxidoreductase that we're going to look at here, it's called lactate dehydrogenase. Now, the starting material or the substrate that it acts upon is this molecule here, lactate, which is the conjugate base of lactic acid. You see that lactate not only requires the enzyme, right? Lactate dehydrogenase is the enzyme. Maybe we should write that in here. This is the enzyme. Not only does it require the enzyme to be converted into the product, pyruvate, but it also needs NAD+. Now, NAD+ is a coenzyme, and as I mentioned, we're going to talk more about coenzymes later on. But what I want you to focus on here is the structure of lactate, right? If we look at what's happening when we go from lactate to pyruvate, how is the structure changing? Well, you can see that we're forming a double bond to oxygen, and we also ended up losing 1, 2 hydrogens. Look, count all the hydrogens in lactate. We have 3, 4, 5. So we had 5 hydrogens here. Now we only have 3 in the final product. So we lost a hydrogen. If we're losing a hydrogen, that must mean that lactate is being oxidized, right? This is undergoing oxidation, okay? This is undergoing oxidation. Now, something else that you might remember from Chemistry 101 is that you can't have oxidation without reduction. It's impossible. They have to exist together. And this NAD+, even though we haven't discussed what the structure of NAD+ is, I'm going to help you out with it. And you can see that NAD+ is changing and it's being converted into NADH. What is that H? That H is a hydrogen. And so the NAD+ is gaining a hydrogen, and so it is being reduced. So it's clear that the lactate dehydrogenase is oxidizing the lactate to pyruvate, but it's causing the NAD+ in turn to be reduced. And down here at the bottom, I just put a little reminder of what oxidation and reduction are in organic chemistry. A transferase transfers a group from one molecule to another. Now, we can break down transferases into some subcategories here, 3 of which are shown here. A transaminase, as you can imagine, if it's transferring an aminase or transaminase, it sounds like it's moving an amino group, right? You're moving an amino group, and that would be completely correct. The next one is a transmethylase. So a transmethylase means you're transferring a methyl group, and I'm sure that everybody here knows what a methyl group is. And then the next one, the name does not really clarify, or it's not really abundantly clear by looking at the name kinase, that it transfers a phosphoryl group. However, kinases are extremely important in this class, and so it's imperative that you know that a kinase transfers a phosphoryl group. Let me show you a reaction that involves a kinase. And this is the first reaction of glycolysis.
I hope everybody wasn't mad at me. I felt terrible. I told Anita, I said, I feel.
So kinases transfer a phosphoryl group. You can see that this is the first reaction in glycolysis, where glucose is converted to G6P, or glucose 6-phosphate. So sometimes we abbreviate this as G6P. Well, what the heck is happening here? You can see that there's a change here, right? This hydroxyl now has a phosphate added to it like that, or a phosphoryl group. And so the enzyme that affects this transformation is hexokinase. The phosphate comes from adenosine triphosphate, which is ATP. If you studied biology, it's for sure you've heard of ATP before. So that phosphate group, or phosphoryl group rather, comes from the ATP and gets transferred onto the glucose. And instead of being adenosine triphosphate, then what you're left over with after you've lost the phosphoryl group is adenosine diphosphate, like that. So this is a kinase, hexokinase, transferring a phosphoryl group, so transferases. Next, hydrolases. Hydrolases cleave bonds by the addition of water. If you think about hydrolase, well, the word hydro means water, right? And in an ase or a lase is breaking, right? Cleavage, okay? So cleavage, cleavage using water. So hydrolases cleave bonds by adding water. And we can break those down into some subcategories as well: phosphatases, peptidases, glycosidases, and lipases. I know it might seem intimidating to look at these names, but if you think about them carefully, you can usually draw or make the connection as to what they do. So a phosphatase removes a phosphate group from a protein. A peptidase sounds like it's got something to do with peptides, and it does. It's the hydrolysis of peptide bonds, right? Remember what a peptide bond is. So if you think about an amide, right, the peptide bond is this bond right here. So that's what a peptidase does. A glycosidase, for example, amylase is a glycosidase that breaks down glycosidic bonds and things like starches. And a lipase will hydrolyze the ester bonds in triglycerides. So if we have a triglyceride like the one shown here, for example, here's triglyceride or a triglyceride being acted upon by a lipase. And it says here it's hydrolyzing the ester bonds. Where are the ester bonds? Well, there's an ester bond right here. There's another one here, and there's another one here, right? This is an ester. Okay, this is an ester. This is an ester. So you're breaking those bonds. What are you going to be left over with? Well, if you look over here, what I have in this green box, you're going to be left over with glycerol, right? After it adds a proton to it, and then you're going to be left over with some fatty acids that come from this part of the ester after it's hydrolyzed. Right there, you end up with some fatty acids. All right, so a hydrolase, again, phosphatase, peptidase, glycosidase, and a lipase. Next, the liase. I said they have something to do with double bonds, didn't I, right? I said liases catalyze the removal from groups to form, the removal of groups, rather, to form double bonds, or they can catalyze the reverse. They can result in the breakage of double bonds. And some examples of liases are things like decarboxylases. They remove a carboxyl group, and a carboxyl group is just, a carbonyl with a hydroxyl from a carboxylic acid. And then another example of a liase would be a synthase, which catalyzes some kind of synthesis process. Now, the example that they have shown in here, I think, is a pretty good one because it makes it abundantly clear. Here we have fumarase, which is the enzyme acting upon fumarate. Well, you can see that there's a double bond in fumarate, isn't there? Right? And we said that a liase can either catalyze the formation of a double bond or the breakage of a double bond. And remember that when you're looking at a double bond, one of the bonds is a sigma bond and one of the bonds is a pi bond. And pi bonds are weaker than sigma bonds. And so the fumarase ends up breaking the pi bond. And what you can see here is that you're adding water across that double bond. That's a concept that we've looked at really from the beginning of the class when we studied alcohols and discussed things like Markovnikov addition. Now, this doesn't have anything to do with Markovnikov's rule because fumarase is acting upon the substrate. It's not an acid catalyzed reaction, but the same concept is here, is that we're adding water across that double bond. So there we go, a liase. Here's an example, or not an example, here's a picture of what fumarase looks like. And the reason I put this in here is just to kind of get us to stop just for a second and to recap, how complex the structures of enzymes really are. So you can see that fumarase is a very big molecule. In the example here, somebody's asking, how do you know which carbon the hydroxyl bonds to or which carbon the hydrogen bonds to? Well, in that case, this specific case, it doesn't matter, does it? Right? It doesn't matter at all because this molecule has a plane of symmetry down the middle. Okay, so if you added the hydroxyl to this carbon or to the other carbon, it wouldn't matter because the two sides are identical like that. Okay, there we go. Let's move on from there. There's the structure of fumarase. An isomerase catalyzes intramolecular rearrangements. Right, we spent a lot of time talking about what isomers are. You might remember that way back in the day, we talked about C2H6O, which we said could either be dimethyl ether, for example, or it could, You can form this isomer from those atoms, which is ethanol. These are two very different molecules, okay, but they are isomers of each other, isomers. And so again, all an isomerase is going to do is catalyze a rearrangement of the molecule. Now, there's a couple of examples or subclasses, sorry, epimerases. These catalyze the inversion of stereochemistry. Say, for example, when we looked at chiral molecules, let's say you had, an R group and then a different R group here, and you had, some kind of chiral carbon. So I'll put a star by this chiral carbon. So we've got a group coming out in the front. Let's say we've got a hydrogen going in the back, for example. What the epimerase will do is it'll reverse those two groups, right? You'll still have your R and your R prime in the same place, but now your X group will be going in the back. It wasn't X prime, was it? And your hydrogen will be coming out in the front like that. And those are two different molecules, aren't they? They are not identical. Those are non-superimposable mirror images. So that's an example of what an isomerase can do, is an epimerization. So we call that an epimerase. Another example would be a mutase. Now, I think the mutases are more common, and these are just converting one isomer into another. For example, we have this SATC molecule here. So this is 3-phosphoglycerate, and the isomerase that acts upon this molecule is called phosphoglycerate mutase, right? We know it's a mutase, so it just converts one isomer into another. If we, look at the molecular formula of this compound, it's got 1, 2, 3 carbons. Okay, it's got 1, 2, 3, 4 hydrogens. It's got 1, 2, 3, 4, 5, 6, 7 oxygens. Okay, and it's got a phosphorus. Okay, if we go over to its isomer 2-phosphoglycerate and we tally up all the atoms, we have 1, 2, 3 carbons. So we have 3 of those. We have 1, 2, 3, 4 hydrogens. Oxygens, we have 1, 2, 3, 4, 5, 6, 7. And then phosphorus, we have 1 of those. And so clearly you can see that these two molecules don't have the same structure, do they? They do not have the same structure, but they are isomers of each other. They have the exact same molecular formula. And so that's what the isomerase does, right? In this case, it's a mutase. It just converts one isomer into another. All right, a ligase. Ligases catalyze a reaction that affects a bond, a specific type of bond, namely carbon-carbon, carbon-sulfur, carbon-oxygen, carbon-nitrogen bonds, or when a phosphodiester is made or broken. So here, for example, this is a DNA ligase, which again must be a ligase, right? And it's acting upon 2 different DNA strands, and it's forming a phosphodiester. So this is a phosphodiester here. Now I know we didn't cover the chapter on DNA, but the rationale still stands for the students in this class, that you all know when you see a new bond being formed, okay, that you should be able to recognize that and say, okay, well, that's got something to do with a ligase. And I think of ligase, kind of makes me think of the word link, like putting things together. So it makes me think of a bond in that regard. Okay, so nomenclature. Again, how do we name enzymes? Well, sometimes it's easy, sometimes it's not. And it depends on the situation. If you're thinking, well, if it's easy sometimes and it's not other times, what kind of questions will you ask? Impossible questions? Of course not, okay? I'm just going to ask you questions that you'll be able to figure out the name by looking at the kind of reaction that it catalyzes. For example, in most cases, enzymes end in the suffix ase, not all the time, but most of the time. The common name for a hydrolase, for example, is derived from the substrate. Now, the substrate in enzyme catalyzed reaction, this is like the reactant, okay? The reactant, or you could call it the starting material. Okay, starting material. Now, in the wet chemistry, right, that we do in the lab, for example, if we took, butane and we burned it in oxygen, okay, we'd say that butane and oxygen are reactants, they're starting materials. We don't use the word reactant when we're talking about enzyme catalyzed reaction. We use the word substrate, okay? So if we think about the substrate that the hydrolase is acting upon, and we just slap the suffix ase on the end, that'll give us the name of the hydrolase. For example, urea is acted upon by an enzyme called urease. What do? We remove the A, kind of, and we just add ASE on the end like that. Lactose, okay, we remove the ose and we get lactase. So that would be the enzyme that acts upon lactose. So if you've heard of, people who are lactose intolerant, which, is a good chunk of the population once you get into people who are older, senior citizens, for example, they lack the enzyme lactase. They don't have a sufficient amount of lactase to metabolize lactose. Other examples of enzymes or other enzymes are named for the substrate and the reaction that they catalyze. For example, lactate dehydrogenase, right? A dehydrogenase must, like, look at it, dehydrogen, right? So, removal of hydrogen. So you must be losing, or you could say removal of hydrogen or loss of hydrogen. What else? Pyruvate decarboxylase, right? If you're decarboxylating something, that must mean loss of a carboxyl, okay? What's a carboxyl? A carboxyl is this, a carbonyl with a hydroxyl, so you'd be losing this portion. Other ones are just annoying, okay? Are just historical names. They have no relationship to any kind of substrate or any kind of reaction. But if you studied biology, I guarantee you that you would have heard of pepsin, chymotrypsin, and trypsin. We usually discuss those in biology. And in fact, we're going to talk about pepsin, chymotrypsin, and trypsin later on in this chapter. So examples of enzymes. Let's move ahead to section 19.2, which discusses that effect the enzymes have on the activation energy of a reaction. And I talked about this earlier tonight. I said that an enzyme serves nothing more than to speed up a reaction by lowering the activation energy, right? Same thing that we talked about earlier on. I'm just going to rehash it here. If we have a reaction or a reaction coordinate diagram like this, and if we have energy on the y-axis, if the uncatalyzed reaction looks like something like this, for example, the catalyzed reaction, all we're going to do is we're going to lower that activation energy. So this would be the activation energy of the catalyzed reaction. So it provides a lower energy route for the conversion of substrate to product. Now, every chemical reaction is characterized by an equilibrium constant. Say, for example, we're looking at a really simple reaction where A is being converted into B. We would define our equilibrium constant as such, right? K equilibrium is going to be equal to the concentration of B to the power of its stoichiometric coefficient divided by A to the power of its stoichiometric coefficient. But this is a reflection of the difference in energy between the reactants and the products. It doesn't have anything to do with the rate of the reaction, right? It has nothing to do with the rate. Now here's that energy diagram again. Maybe this is a little bit clearer than what I wrote out. This is from a reaction where we would have A plus B, giving you C plus D, for example, okay? We have our uncatalyzed reaction. What's characteristic about it? It's got a really large activation energy, whereas our catalyzed reaction, right, our enzyme catalyzed reaction has a small activation energy. In the catalyzed reaction, in the above right, the activation energy has been lowered significantly, and what that does is that increases the reaction rate, the rate of the reaction. Well, how much is it going to affect the rate of the reaction? Now, in chemistry that's, wet chemistry that we do in the lab, we kind of have a rule of thumb. Students love rules of thumb, and a rule of thumb that we use in general chemistry is that if we have a reaction that we need to heat up, for example, every time we increase the temperature of the reaction by 10 degrees Celsius, we usually see a double increase in the reaction rate. Now, there are exceptions to that, but what we're going to see is that enzyme catalyzed reactions don't work the same way, right? Organic chemistry is very different from general chemistry. So let's take a look here. It says rates of uncatalyzed reactions increase as the substrate concentration increases. Think about it. If we have some enzymes, right? We have a whole bunch of enzymes that can affect some certain transformation, okay? Well, if I have a lot of enzymes and I have, a little bit of substrate or reactant that's going to react with those, okay, well, think about it. The more of the substrate I have, well, the faster the reaction's going to go, right? Okay. But what it says here is that the rate of the enzyme catalyzed reactions show two different stages. Now, this is very different from what you saw in Chemistry 101. It says here the first stage is the formation of an enzyme substrate complex. You're like, what the heck is an enzyme substrate complex? Well, let's think about it. In order for the substrate to be converted into product, it's going to have to actually interact, connect, bind with the enzyme itself. Okay, we're going to talk about that in a lot of detail. This is followed by a slow conversion of the substrate into product. Now, what the enzyme is going to do is it's going to affect the transformation of that product, or sorry, that reactive, that substrate into the product. Well, here's the kicker. Okay, so this is what's very different from general chemistry, is that the rate is actually limited by enzyme availability. Now, if we were to take, an old reaction that you would have learned, say, in general chemistry, again, what the rule that we learned in if you studied, Chem 101, for example, at PPCC, what you said is, okay, well, if you increase, the concentration of reactants, right, your substrate, the more you increase it, the faster the rate's going to go. Okay, and that's what we see here. But when we're dealing with an enzyme catalyzed reaction, so this is in Chemistry 102, we see something a little bit different. At first, if you just, if you were to just look at this part, right, of the graph, what does it look like? Looks like a linear slope to me, man. Okay, it looks like a beautiful linear slope. So you'd say, hey, it's just going to keep on going and going and going and going and going like that. Hey, the more substrate that I add, the faster the reaction's going to get. But what you see is that's not the case at all. Okay, you get to a certain concentration. Okay, you get to this concentration where is it right around here, and all of a sudden it just flatlines, right? The rate of reaction doesn't increase. You can keep cranking this up, right? You keep cranking that up, but the reaction rate stays the same. Now, what's that all about? What's that all about? Well, we're going to explain why that is. Why, when we're dealing with enzyme catalyzed reactions, there seems to be a point there where the reaction rate ceases to increase. And what that has to do with, it has to do with enzyme availability. Now, we don't have an infinite number of enzymes, do we? Okay, so again, going back to Chemistry 101, we're talking about uncatalyzed chemical reactions. Oftentimes, they're going to double when the substrate concentration is doubled. Although the reaction was initially responsive to substrate concentration, wasn't it? At a certain concentration, it just stopped, right? The reaction didn't stop, but the rate reached a maximum value. And that's what happened right here, okay? That's what happened here, okay? At first, it was responsive, right? It was responsive here, responsive to increases in concentration. But then after a while, it died off. And then after a while, there was no change whatsoever. So that's the point, okay? Now, again, the rate of the reaction initially, it just was skyrocketing, wasn't it? But then it leveled off at a maximum value. Now, here's the explanation. At its maximum value, all of those active sites where the substrate can interact or bind with the enzyme, they're all filled up, okay? If all of the enzymes you have, and you're limited by the number of enzymes you have in your living organism, if they're all filled up by a substrate molecule, well, you can keep throwing molecules at it, but they're not going to be able to bind, are they? So the answer is this: you're limited by the amount of enzymes that you have. It's a finite amount. And so once they're all filled up, the reaction is just going to taper off, right? It's going to reach that maximum value. Now, there probably are some explanations that I could give, or some examples. One that I tried to use as an illustration before, I don't know if it's the best illustration in the world, but you can think about it like this. If you had a parking lot, okay, if you look at, say you went to Walmart's parking lot, right, and there's all kinds of parking spaces. Well, if you were to go to Walmart on a busy day and you see that you watch, you count the number of cars that park every, minute or something like that. You'd probably see that, during the peak hours, it's kind of like a certain rate that is, it increases or whatever. But at a certain point there, once all the parking spaces are filled, there's not going to be any more people parking, right? So initially, the amount of people parking and parking is going to increase, increase, increase. But as the spots get limited, and then eventually, when they're all filled up, well, nobody's going to be parking at all. I don't know if that's the best illustration in the world, but hopefully, it gives you some kind of something to hang on to there. All right, let's talk a little bit more about that enzyme substrate complex. Again, enzymes are really complicated molecules, right? If we go all the way back to the beginning, okay, let's go back to the very start where we had this, complex structure of an enzyme. All of this thing, okay, this is the enzyme. So the enzyme is really big. There's only a small portion where you're going to have what's called a binding site, okay? So the big part, this is the enzyme. Enzyme, huge, huge molecule. But this little molecule in here, that's the substrate. Substrate. So that entire gigantic molecule, that enzyme, exists just to act upon this little tiny, teeny, weeny substrate that actually fits in a very specific spot. And if you're saying, Mr. Dion, that looks just totally random. It looks like spaghetti and somebody threw a molecule in it. It might look that way, but I can guarantee you that the structure of an enzyme is very carefully put together. Evolution designed these to have a specific sequence of amino acids and to have specific secondary, tertiary, and quaternary structure. Okay, so the substrate is actually going to very specifically always bind to the same place in the enzyme. Okay, now if you're like, well, what the hell's holding it in there? Is it tape? Is it duct tape holding it in there? No, there's going to be some intermolecular forces that must be holding it in there, right? Okay, well, let's back up to where we were and talk a little bit about that enzyme substrate complex. So these reversible reaction steps, and you can see there's 4 of them, step 1, 2, 3, 4. Okay, these reversible reaction steps represent the steps in an enzyme catalyzed reaction. Look, if you go back to chemistry 101 or chemistry 102, even when we talked about esterification or, hydrogenation or whatever, okay, all of those reactions are just A to B, right? Like, say, for example, I'm trying to come up with a simple example off the top of my head. Say we took something like ethylene, right? CH2, double bond CH2. And then we did a hydrogenation reaction with platinum, and then we ended up with ethane. That's a hydrogenation. That's just A to B, right? Okay. Well, enzyme catalyzed reactions are a little more complicated, okay? They're a little more steppy. So we have all of these different steps. Let me walk you through them. Okay. Just like that song, Some Enchanted Evening, when you spot your true love across a crowded room, the enzyme and the substrate see each other, right? Now, obviously, they don't have eyes, they're not seeing each other, but you get my point, is that they must be in proximity to one another. Okay, so they're in the same vicinity. Okay, so we have the enzyme and the substrate, and what's going to happen is that substrate is going to bind to that to the enzyme, and we get what's formed. We get what's called a complex. Now, notice that I always say the word bind. You notice that I never say the word bond. Okay, I didn't say the enzyme bonded to the substrate. Why? Because that would imply that you're forming a covalent bond between the substrate and the enzyme. When you form a covalent bond to an enzyme, that's usually what we call an irreversible binder, and those are usually things like poisons, like nerve gases, okay? So we're not talking about that. We're talking about binding. How does the substrate bind to the enzyme? The good news is that you covered all of these ways that the substrate binds to the enzyme. We covered all these in chapter 18. Okay, we talked about things like hydrophobic interactions, right? Hydrogen bonding, salt bridges, things like that. So it's going to be intermolecular forces, not bonding, but intermolecular forces, okay, that are going to hold the substrate to the enzyme, okay? Now you've got them together, right? They're happy as a clam, right? E plus S is now ES, two become one, right? So it's an old Spice Girls song. Well, what happens here is I told you that at some point you're going to end up with a product, aren't you? That's where we're trying to get to. We're trying to get to the product. While the substrate is bound to the enzyme is going to do something to it. And if you're like, well, Mr. Dion, you're being kind of vague, just saying it's doing something. Well, it depends on what the enzyme is. Okay. But let's say it's a ligase, okay, and it's cleaving a bond. Oftentimes, what a ligase will do is it will simply put stress on that bond. If you're like, well, how the heck is it going to do that? Remember that protein. When we talked about these big proteins, we said that they didn't have a rigid shape, did they? Okay, they had a well-defined shape, but it isn't rigid like a stone, okay? There's some flexibility. So what happens is that when the substrate binds to the enzyme, that enzyme might flex, and this is just a specific example, but it might flex in such a way that it's going to take that substrate and it's going to put some stress on a bond in there, okay? So much stress that it's going to snap that bond. Okay, well, when that bond is broken, you don't have the substrate anymore, do you? You don't have the starting material. Now you've got a complex between the enzyme and the product. So once the enzyme has converted that substrate into the product, it's like, I'm done. My work here is done. Be free, product. Leave, fly, do your thing, okay? So then it releases the product. Okay, give me a thumbs up if that makes sense to you, or let me know if you have any questions about that, because those are important concepts for you to understand in this chapter for us to really, plow on. Good, Madison, good. All right, great. Okay, yeah, great, Ryan. These are really important concepts. The whole formation of an enzyme substrate complex, then a transition state, enzyme product complex, and then release of the product. Okay, good. I'm glad you're on the same page as me. Well, let's talk a little bit more about the enzyme substrate complex, right? The E plus S. The enzyme does change shape, okay? It doesn't change shape in that it's a complete, an utter change of the opening or something. Okay, what you're going to see is that there's going to be subtle changes, okay? Subtle changes in the molecule, right? What is an enzyme? It's just a really big molecule. And we know that molecules have bonds and bonds can move, right? Bonds are not rigid. There's going to be some rotation in there. And so remember, this is something that we learned in chapter 18, that proteins are not rigid, right? Their conformations can change. And so an enzyme's conformation can change. And we're going to look at that even more later on when we talk about the lock and key model versus the induced fit model for enzyme catalyzed reaction. All right. So what you're going to see, Madison, is that when the substrate binds to the enzyme, okay, oftentimes the enzyme will change its shape to some degree. I would say more of it's a change in conformation to affect the reaction. After the reaction is done, it'll go back to its original form. All right. Okay, let's talk more about the enzyme substrate complex, right? We said it was ES, right? That were E and S together, right? That's the enzyme substrate complex, okay? The part of the enzyme, remember the enzyme is a big structure, right? It's a very big molecule. But that little part of the enzyme that combines with the substrate, where it binds, it's got a specific name. It's called the active site. Okay, that's where the magic happens. Active sites include these characteristics. They oftentimes have pockets, right? What's a pocket look like? Okay, it's just a pocket like that. Okay, it's going to be some kind of spot where the substrate can fit inside of a pocket, or a cleft, which is another way of, kind of describing a pocket. So a pocket or a cleft in the surface of the enzyme. And what's interesting is that the R groups in the amino acid residues or portions, we could call them residues in the enzyme. So where the active site is, right? Remember, the enzyme is made out of amino acids. So all the R groups that are in that pocket, maybe I could draw the pocket a little bit bigger. Okay, say we have a pocket like this. The R groups that are on the inside of that pocket, and they're, different, okay, whatever they are, okay? All those R groups are going to interact with the substrate, okay? And they're going to interact with that substrate in what kind of ways? So first of all, the shape, okay? The shape of that binding pocket is going to be complementary to the shape of the substrate. You see how the substrate I drew is just some like blue oval that kind of fits this oval shape. If the substrate was, for example, and I'm being ridiculous here, but if the substrate was a triangle, it wouldn't fit in there very well now, would it? Right? So the shape will be complementary for one thing. Okay. Also, the enzyme is going to hold and attract the enzyme using non-covalent interactions. What are these? Our favorites, van der Waals forces, okay, like London forces, dipole-dipole attractions, and electrostatic interactions, right? Things that we learned about before. Hydrophobic interactions, hydrogen bonding, salt bridges, things of that ilk. That's so the R groups that are on the enzyme, right, are going to interact with those R groups, and that's what's going to hold it in here. Remember, this is not covalent bonding, right? These are intermolecular forces, IMFs, that are going to hold that substrate in there. Now, the conformation or the shape of the active site is going to determine the specificity of the enzyme, okay? So remember, not any substrate is going to react with any enzyme. It doesn't work that way. Some enzymes are more promiscuous than others, meaning that they can react with a wider variety of substrates, but there are some enzymes that are very substrate specific. Here's an example that might, or a picture that might answer your question a little bit more, Madison. So here's our big old enzyme. Okay, there's the enzyme. You can see that the binding pocket or the active site is right here. Okay, so that's our active site. Now, it's going to bind to our substrate and it's going to form the enzyme substrate complex. Okay, so, I kind of think of it as going in here and it docks in there, and you see how it actually fits really well. You see how it holds kind of the contours of the substrate, and where those green markings are, that represents the intermolecular forces, okay, that are holding that substrate in there. Now, after the enzyme has done its magic, okay, and let's say, for example, it's, breaking apart this substrate, again, it's going to do something to split up the molecule, isn't it? But once that's done, it's going to release it as products. And now we've got here, this is the enzyme product, okay? I should say enzyme product, but anyhow, it's the enzyme substrate complex, and then it releases the products. Okay, anyhow, two models have been proposed to explain what the heck is going on here. This whole substrate binding with the enzyme. Let's take a look at these two models, and you're responsible for knowing these, okay? So all Chemistry 102 students have to know what the two models are. I think they even come up on the final exam. So let's get into them. The first one is the lock and key model. This is an old model. Do you remember Fisher, Emil Fisher, who developed the Fisher projections that we looked at in the carbohydrates chapter? If you're like, what the heck is the Fisher projection? You remember when we drew, carbohydrates like this, okay? Like that, and then we had all the horizontal lines like this. That's called a Fisher projection. So the same Fisher, Emil Fisher, back in 1894, he proposed the lock and key model, and it's this, okay? He said the enzyme and the substrate are made to fit each other exactly, okay? It's a perfect fit. It's like a hand in a glove. If you look at the example shown here, you see how the contours of the enzyme exactly line up beautifully with the contours of the substrate. Okay, it's a good model, right? In fact, when I worked in industry, and we had people from the government or something who came to visit, and we were begging for money for research or something, the way that we would describe enzymes is that we would use a lock and key model, okay? Because it's very easy for people who haven't studied chemistry before to understand. We just say, okay, we've got an enzyme, it's like a lock, and we're trying to find, the key that's going to fit that lock to, prevent this reaction from occurring that causes cancer cells to metastasize, right? Okay. Well, the problem with this model is that it fails to take into account that proteins can undergo conformational changes, okay, in order to accommodate a substrate molecule. So a much more accurate representation is what's called the induced fit model, enzyme model. But before we get into that, let's look at another example of the lock and key. This is basically a rehashing of the slides that we looked at a couple of seconds ago. So the active site in the lock and key model is rigid. Okay, active site is perfectly rigid. You can see here it is right here, and it doesn't change here, and it didn't change here neither, okay? So that key, that substrate fits in there exactly. It gets released. Okay, there we go. So let's take a look at the induced fit model. Again, more accurate, more modern. Was discovered in 1958. So what it says is that the induced fit model of enzyme action assumes that the enzyme active site is more a flexible pocket whose conformation changes to accommodate the substrate molecule. Now you see here that the contour of the substrate, which is kind of like a hexagon and a pentagon here. So I think it's supposed to represent sucrose, because sucrose is made from glucose and fructose, and the 6 and 5 membered rings respectively. Anyhow, you can see how the contours of the enzyme, there's two pockets, but they definitely don't fit exactly. So what the enzyme does is it's not going to change its structure completely, but it's going to be flexible enough that the binding pocket, okay, can flex or change its conformation a little bit so that it's going to line up, and fit better with your substrate like that. Okay, so that's the induced fit model, which is a more accurate model of how enzymes bind to, how enzymes bind with their substrates. But just to put this bug in your ear, before the days of coronavirus, when I gave paper exams and my students would get essay questions, oftentimes I would give them some form of an essay question that said, explain the induced fit model and the lock and key model, and give me an example of how they work, kind of thing, something like that. So here's another pictorial representation of the induced fit model. Now I want you to pay attention to this very carefully. That you see here's the pocket, okay? But look what happens to this portion right here of the enzyme. Okay, once the substrates bind, it might not be easy to tell, but you see how it's kind of on this angle here? Look how it kind of curls up here. So it actually flexed, and then it goes back to being straight again when it releases the product. What does that mean? That means that more substrates are now ready to fly in there, right? So it undergoes a conformational change. Remember, it's flexible. It's not changing completely, entirely. It's flexible enough that it can fit the substrate into the cleft or the pocket, so to speak. Okay, let's talk about the specificity of the enzyme substrate complex. Right, you have a substrate, maybe it's in the human body. You have an enzyme. Well, how do we know that the enzyme is going to act on it? And what about the enzyme? Why doesn't it react with everything? Why doesn't every enzyme react with every substrate? Well, as I told you, it depends on the type of enzyme. For the enzyme and substrate to react, the surfaces must be complementary to one another, right? They have to have some kind of complementary surfaces in order for the substrate to bind, right? If the R groups on the enzyme don't align or match up with the R groups on the enzyme, well, why would they bind? Okay, now a little more about the specificity of enzymes. Enzyme specificity is the ability of an enzyme to bind to only one or maybe just a very few number of substrates. Now here's a very specific example, and this is urease. You can guess what urease hydrolases, right? Urease is a hydrolase, and hydrolase because it's using a reaction with water here. But anyhow, urease acts on urea that has a very high degree of specificity. Okay, high degree, and I even capitalized it. That means that it only reacts with one molecule. Okay, one stinking molecule, the urea molecule. Okay, this is the first organic molecule ever synthesized by human beings. Right, Friedrich Wöhler made urea from inorganic starting materials. Anyhow, urea gets acted upon by urease to make carbon dioxide. And what's this molecule here? Ammonia. Ammonia is very stinky. Okay, and that's why urine smells, because urease acts upon it to form ammonia. Look at this. This is methylurea. Not that different from urea, is it? It looks pretty similar to me. The only difference is it's got this little methyl group on it here. But if you react methyl urea, even though it's very, very, very close in structure to urea, you react it with urease, nothing happens. Zilch. That means that urease is so specific that it only reacts with this one molecule. So a very high degree of specificity. There are classes of enzyme specificity, and urease would fall under this category, absolute specificity. That means that the enzyme only reacts with one specific substrate, nothing else. It's just a one-trick pony. Okay. The next one is group specificity. Well, you can probably guess that it has something to do with molecules that have the same functional group. So maybe it's going to act upon the ester group of the triglyceride, or maybe the amide in a protein. Okay, so functional groups. The next one is linkage. You could probably guess that it has something to do with a certain type of bond. So maybe it's going to only break, a carbon-nitrogen bond, or a nitrogen-hydrogen bond, whatever. Okay. And the last one is stereochemical specificity. We spent quite a bit of time in the carbohydrates chapter talking about stereochemistry. Remember that enantiomers are non-superimposable, mirror images of each other. Okay, well, if I have two non-superimposable mirror images, what you're going to find is that an enzyme will bind or react with one enantiomer, but it won't react with the other. What's cool about stereochemical specificity is that virtually all of the enzymes in the human body have stereochemical specificity. So when you make a drug, if you're a chemist and you're designing a new drug that's going to go into a human being, you not only have to take, the structures or the structure and the connectivity of the atoms into account, but you also have to take its stereochemistry into account. So it definitely adds a layer of complexity to synthesis of organic molecules. All right, let's talk a little bit about transition state and product formation. So how does the enzyme promote a faster chemical reaction? Well, as the substrate interacts with that enzyme, its shape changes, and this new shape is energetically less stable. So like I said, the enzyme is flexible, isn't it? But the enzyme is much, much bigger than the substrate. So when the enzyme changes its shape, okay, it's going to change the shape of the substrate, and it's going to change it to something that's less energetically stable. If it's less energetically stable, what does that mean? It's going to break it apart, okay? It's going to cause it to change into something else, okay? This transition state has features of both the substrate and the product, but it falls apart to yield the product, which dissociates from the enzyme. Well, let's look at some ways that the enzyme might cause the reaction to occur, okay? So what we're going to do in the next few slides, I want you to pay attention very carefully here. For some reason, okay, the enzyme is affecting a change in the substrate, isn't it? Now, what exactly is the magic that the enzyme is doing to that small molecule to get it to convert it into something else? Or maybe it's two small molecules that are being joined together. Why? How? How is this happening? Well, first of all, if you're taking if you're starting with one molecule and you're ending up with two molecules, well, in that case, it must be breaking something, right? So in this case, the enzyme is going to be putting enough stress on a bond that it's basically going to break it. Nothing more than that. So you're just going to end up breaking that bond right there. And then it's going to release two products, okay? This is another cartoon that represents what was on the last slide. I just took it from a different textbook. I thought it was a good figure, to help my students understand what the hell is going on. This is the hydrolysis of sucrose, which is the substrate. The sucrose is the substrate. Right, sucrase ends in ase. This is the enzyme. Okay, well, what the heck is happening? Here's our sucrose molecule. So this is the sucrose molecule here. This is the sucrose enzyme, okay? The sucrose is going to approach the enzyme, then it's going to bind, okay? It's going to bind to the active site. Then water is going to hydrolyze, right? Water is going to attack it, and it's actually going to break that bond in order to release glucose and fructose. Once they're liberated, what do you have left over? You've got the enzyme, and it goes right back to the beginning, and it's ready to act upon another molecule of sucrose. Another possible transition state change. Here's an example of where we start with two substrates, and we bring them together to form a new molecule. Okay, so we're going to start here with number 1, where I have the red circle. We have the two molecules that are close together. Now, they both kind of look like the Rudolph the Red-Nosed Reindeer, right? They have a red nose, okay? But what those red dots are representing, those are the functional groups that are going to come together, okay, to form a new bond. So look, the functional groups, when the substrates bind to the enzyme, look at how crazy amazing nature is, okay? They don't just bind any old way. Nature knows that those two molecules have to bind next to each other in such a way that these two functional groups are aligned. Okay, so you think, oh, they could just, bind any old way. No, no, no. It's very specific about how it binds to the enzyme. So once the two functional groups approach each other, you form the bond, you lose water, and then you end up releasing the product. What are you left over with? You're left over with the enzyme that's ready to start over again. Now, again, what's being shown here, instead of sucrase, which catalyzes the hydrolysis of sucrose, this is showing sucrose synthase, so the formation of sucrose. If you look at all the hydroxyls on fructose and all the hydroxyls on glucose, because it's two hydroxyls that come together, right? When you lose water to form the product, there's literally a 1 in 25 chance that the two correct hydroxyls are going to be aligned like that. But again, evolution has designed the enzyme such that it knows exactly which hydroxyls have to be aligned next to each other in order to form that new molecule. The third one, or the third possibility here for a transition state change, is that the enzyme might modify the pH of the microenvironment. The microenvironment just means the environment around the substrate, and then it can cause, the donation or the accepting of a proton, which will catalyze, catalyze the reaction. So nothing more than that. All right, that brings us to section 19.7, cofactors and coenzymes.